Okay, apologies everyone. We had a few technical details that we were just trying to sort out, so I won't be able to use the wireless microphone, but I will be able to use the podium microphone, so I'm going to have to stick around the podium, which is not my favorite thing. So just a couple of quick administrative notes to make sure that we're all on the same page. The first and most important thing is that your term tests are always going to be on a Monday during class time. You will not have a lecture afterwards, so it's designed to be like 1 hour. You'll get the full 60 minutes. You'll show up to the exam center or whatever rooms HMB has booked for you, and then there won't be a lecture afterwards, even if you end early. So the other thing to make note of is if for any reason you are not able to write the regular sitting, there is a make-up term test. You can follow sort of the procedures that are outlined for you on the course syllabus. However, I can also tell you that the format is different, but this exact same material and this same level of difficulty will be covered on any make-up term test, but I just wanted to make that clear for everyone as well. So if you don't have any other questions or anything that I can clear up for you now, we'll just get started with today's lecture. Do you have any questions? No? Okay, so we're going to get into a couple of different aspects of different things that we started talking about last week. You need to actually pay a lot of attention this week because this material will be very heavily emphasized on your term test. We are recording the lecture, so you will be able to go back and re-listen to it. And during this time where we have enrollment continuing on all the way till Sunday, we'll continue to record both the Monday and Wednesday lecture. But I am kind of evaluating what the attendance is going to look like. So again, when we're looking at neurobiology, there are a lot of really cool things that we're going to talk about later. How do you actually get an animal angry just by flashing light in its brain? And so we'll talk a little bit about optogenetics, another technique that we'll learn about later. But the fact is, every single technique that we look at within this course really relies on the fact that you understand how neurons signal. How do these different receptors, these channels, how do these different biophysical properties of changing the membrane potential that we're going to begin today, how does that actually make a difference in turning an animal angry or making the animal fearful or making the animal less fearful when we look at things like optogenetics? So I need you to understand the basics before we can get into some of the other really cool things that we'll look at later on in this course. So in the last lecture, what we looked at was what are some of the basic properties of these channel proteins? Charged particles, the things that are actually carrying charge that will change the membrane potential as they go from one side of the membrane to the other. They can only do so if there is a channel protein that they can use to cross that membrane. Charged particles by themselves cannot go across a lipid bilayer any more than I can walk through this wall. And so we need a channel protein. And we looked at different things that affect these channel proteins. And at the end of the last lecture, and on that orange sheet, there are some questions about open channel blockers. We looked at some of the different properties of these different channels. One of the things that we didn't cover, although we looked at it last week, is what is the function of these ligand-gated channels? How do we get a neurotransmitter to open up a channel that is part of a receptor? So I'm going to use those terms interchangeably, and many of your neuroscience profs will also use these different terms interchangeably, a receptor and a channel, but I'm hoping that you'll understand based on the structure of the different receptors that I'm going to show you today that indeed there is a channel, there is a physical opening. So if we're looking at it not from the side, but we're looking at it from the top, you can actually see that there is a pore or a hole in which ions can traverse if this pore were to be able to open. And the next 5 or 6 slides actually deal with how do these different channels open in response to an appropriate signal. So one of the best studied types of receptor channels is the nicotinic acetylcholine receptor. So this is the first one that was studied in any length or in any detail. And again, hopefully now you understand when I talk about a receptor or when I talk about a ligand-activated channel, you understand why all of these different receptors have channels built into them. We see these often within the muscle, particularly in the postsynaptic compartment. So we're not only looking at this in the brain. We do see these on neurons, but most of the time, when you see nicotinic acetylcholine receptors, you often see them associated with skeletal muscles, and you see how skeletal muscles can become activated, but we do see the exact same thing in neurons as well. So again, skeletal muscle or nerve fibers will have these postsynaptic acetylcholine receptors. These are, again, when they are open, when we get this pore to form inside of them, these become permeable to cations. And again, all channels, no matter what we're looking at, they rely on the fact that they're always passive. They will always just conduct or allow ions to flow through along their concentration gradient or down their concentration gradient. So if we are permeable to cations, that means that we need to have a concentration gradient for these ions to be able to flow through. For the nicotinic acetylcholine receptor, the complex as a whole, we actually see that it consists of several different subunits. So this is the entire nicotinic acetylcholine receptor, but the entire receptor actually consists of an alpha subunit, in fact, 2 alpha subunits, a beta subunit, in this particular case, a delta subunit, as well as a gamma subunit. So we have 5 of them. They're not all the same, so we don't have 5 alphas together. We don't have 5 betas together. We don't have 5 gammas together. So we call this a heteropentamer. So 5 subunits, that's the pentamer part. The hetero meaning that we have different subunits that have to come together to form this nicotinic acetylcholine receptor. And the different types or the different amounts of the alpha versus beta versus delta versus gamma, we call this receptor stoichiometry. So if we have 2 subunits of alpha, 2 subunits of beta, and then 1 of gamma, that tells you about the receptor stoichiometry, the overall ratio or the numbers of these different subunits. Typically, in order for a nicotinic receptor to work, we need to have 2 alpha subunits. The importance of this alpha subunit is really the site of where acetylcholine binds. So if acetylcholine can't bind, this receptor won't be able to open. Okay, so again, keeping that in mind, this is the basic structure of a heteropentameric nicotinic acetylcholine receptor. And now I'm just going to go back and give you a big picture of what all of this means. So acetylcholine is a neurotransmitter that we use in the nervous system. We also use it in the skeletal muscular system. And when it is released and when it binds to the appropriate acetylcholine receptors, what I've just indicated to you are the nicotinic acetylcholine receptors, and these are true ion channels. We also see that we have G protein coupled receptors, which we call muscarinic receptors, that involve a different pathway. And again, this is a very common theme. When we have a neurotransmitter, in this case acetylcholine, often we have both an ionotropic receptor, one that allows ions to flow through, and then another one that is distinct and separate, which are, again, using the same neurotransmitter, but then activate a G protein and allow for a signaling cascade to occur. So these are not channels when we talk about these muscarinic type. We only see channels associated with the nicotinic type, and we identify the differences based on the pharmacology. So nicotine will also bind to acetylcholine receptors, and the chemical substance known as muscarine will only bind to the G protein coupled receptors that are involved in the nervous system. So again, another really important slide. So the previous 2 slides are important. This slide is also important. And when I ask, or if I were to ask you to draw the topology of each of the individual subunits, it turns out that if we're looking at the alpha subunit, or the beta subunit, or the gamma subunit for the nicotinic receptor, they all have the same topology. It doesn't matter what we're looking at. Alpha is the same as beta. They all have this overall topology. And the topology, if you were asked to draw it, is better to draw it in the simplified version, which is what you see here. So what we see is there's this really long and very big extracellular N-terminal region. So this is the N-terminus. And then we have one transmembrane domain that you see here, a very small intracellular loop, which is inside the cell. Then we have the second transmembrane domain, which is joined to the third by this very small extracellular loop here. And this third transmembrane domain, which some books will call M1, M2, M3, M4. And in this diagram, it's called TM1, TM2, TM3, TM4. It's all the same thing, just means membrane or transmembrane domains. And we can see that these 4 domains, and here between M3 and M4, we have this really large cytoplasmic loop. Regardless of what subunit we're looking for, whether it's alpha or beta or gamma, delta, any type of nicotinic acetylcholine receptor, all of the subunits have exactly the same structure. And it turns out that each of these transmembrane domains is important for different types of things. But the most important one that we're considering is the M2, or the second transmembrane domain, because this is the domain that actually forms the channel. So if we were to take a look, and I'll show you this on the next diagram, but if we were to look at this channel here, the inner portion of this channel would actually consist of the walls of the M2 subunits that are going to be aligned to that channel pore. And if it were to open, then we would see cation ions like sodium flowing down their concentration gradients and allowing for depolarization to occur. And again, you can sort of see the heteropentameric structure, each of these subunits. And if we were to take one of these subunits, this is what it would look like in terms of its topology. So because this is an important slide, and I need you to understand this because GABA A receptors are identical to this, do you have any questions that you are comfortable asking now before we go on? Yes?
So when you say that the pore region is like of the walls of the M2, do you mean like between the walls of the M2?
No, the actual transmembrane domain itself. So you'll have to imagine that each subunit, one of the M2 transmembrane domains will form part of the wall. Okay? Any other questions? Yes?
One alpha subunit enough?
No, you always need 2. So 2 alpha subunits are required for the activation of the nicotinic acetylcholine receptor. Any other questions? And again, just to show you this in a slightly different way, I would never ask you to draw the figures that are here on the left, nor would I ask you to draw the figures that are here at the bottom. It's just to give you an idea of what we're talking about in a slightly different way. So again, this is one subunit of the nicotinic acetylcholine receptor. And if we were to take a look, the M2 region, which is shown to you in 3 dimensions here, is the one that actually forms the pore region that you see here. Another way to take a look at that would be to see what happens here. If we were to take a look, the blue dot represents the M2 pore region, and what we're seeing is that they all rotate to open when the appropriate signal is being applied. In this case, we need 2 acetylcholine residues to bind to the alpha subunits. When they bind, these M2 regions will then rotate and form the walls of the acetylcholine receptor channel pore. I'm hoping that gives you sort of all the information that we just talked about in a slightly different form. So again, because this concept is an important one, that the M2 region forms the pore that will allow sodium to flow down its concentration gradient. Do you have any questions before we go on? No? Okay, again, keeping in mind that you do need to know the topology, the stoichiometry, how these different subunits all come together. So this was actually one of the best studied receptors for a very long period of time, and it became the model system for many of the receptors that we look at within the nervous system. One of the other types of receptors, the inhibitory receptor type, are known as the GABA receptors. Just as I showed you earlier with acetylcholine, GABA itself can be separated out into G proteins as well as ion channels. If it is a ligand-gated ion channel, we call this a GABA A receptor. If it is a G protein coupled receptor, so this is on neurons, and I'll show you that in a moment, then one of the things that we know is that we have a GABA B receptor if it is a G protein coupled receptor. In fact, unlike some of the other G protein coupled receptors that you might have learned about in pharmacology classes, this G protein coupled receptor actually exists in a dimerized form. We actually have two G protein coupled receptors that have to work together within the nervous system, and this can consist of GABA BR1 or GABA BR2. And again, I'll show you that in the next slide when we get to it. So the other thing to make note of, when we have this heteropentamer, so very much like the nicotinic acetylcholine receptor, we have 5 subunits that have to come together. Here, we have a lot more diversity. GABA is one of the more important neurotransmitters that we'll be looking at when we look at epilepsy and when we look at how different types of seizure activity occurs, when we have different types of inhibition issues. We will look at this GABA A receptor, and it turns out GABA A is incredibly important, so we have more diversity. So not only do we have alpha subunits, beta subunits, gamma subunits, delta subunits, etc. In fact, we have multiple families. So we have alpha 1 or alpha 2, alpha 3, all the way up to alpha 6. And for the beta subunits, we could have beta 1, beta 2, beta 3. That means that we can mix and match many, many more types of these different GABA A receptors than we would have for the nicotinic receptors. Nicotinic is just alpha, beta, and we'll keep it simple, but GABA A receptors are much more complex. One of the biggest questions that we've had in neuroscience is, when I just showed you that diagram of the rotation of the M2 domain, and it's identical for the GABA A receptor, how does this actually open up the channel? And it was actually hypothesized for a very long period of time, and it wasn't until early January of 2019, so about 2 weeks ago, there were a series of back-to-back papers that came out in Nature, and again, you don't need to worry about memorizing any of these, that using very, very specialized techniques that they developed themselves, were able to show that this rotational behavior that we had predicted with the nicotinic receptor that I just went through with you actually does occur, that when GABA binds, and here I need you to understand that the GABA binding site is a little different. For acetylcholine, acetylcholine binds to the alpha subunit. For GABA receptors, it binds at the intersection of the alpha and beta subunits. So unlike acetylcholine receptors where we have to have alpha, for GABA, we need to have both alpha and beta subunits for GABA to work on this ionotropic receptor. And again, it wasn't until this paper came out that showed incredibly elegantly using very, very high resolution mapping and different forms of mega body antibodies that I'm not going to spend time getting into now, that they were able to show that this mechanism that was proposed 20 years ago, they could actually view this occurring. So this theory is actually a really solid theory, and we now accept that there is this rotational behavior when the ligand, in this case GABA, binds to the appropriate site. So one other question to think a little bit about. I didn't show you this, but you should make an educated guess on this. If you're looking at the overall topology of these receptors, and I'll show you the GABA A receptor subunits in a moment, of all of these different domains, where do you think the neurotransmitter is going to be binding? Is it going to bind to a transmembrane region? Is it going to bind to the cytoplasmic region? Where do you think it would most likely bind? Yes? Probably the extracellular N-terminal region, which is this massive domain that you see out here. And it turns out that that prediction is also correct. So just again, before I go on, and you do need to know this particular slide, so just again to make sure that you are aware of it. So the data or the information on this part of the slide is important. And again, we didn't talk about the movement of chloride in great detail, but we will starting later this week or early next week. When we look at the GABA B receptor, it turns out, again, on this paper that was just published last week, that these GABA B receptors are also incredibly important. They regulate the release of neurotransmitters. It also answers this really overwhelming question that we've had in neuroscience for a really long period of time. Why do we have amyloid precursor protein? You might have asked yourself that. So if amyloid beta, that toxic fragment that we talked about in lipid rafts exists. And if it's so bad, why do we have this around? And why does everyone have it on their cell membranes, especially around those lipid raft regions? And it turns out that that cleavage of that amyloid precursor protein actually might be working directly on these presynaptic GABA B receptors. And I'm going to go through this in a little bit more detail. So you might want to go back and review what we learned about for lipid rafts, amyloid precursor protein, how it uses the BACE or BACE, the beta amyloid converting enzyme, as well as the gamma converting enzyme to actually form this soluble protein, or A beta, when it gets cleaved more, and why this soluble amyloid precursor protein might be interacting with the GABA BR1 unit. Remember, we have a dimer, and so you can see this G protein coupled receptor dimer. So we have two of these G protein coupled receptors together. This is very unique in terms of the nervous system. And when this soluble form of amyloid precursor protein binds to what is known as the sushi domain, so it is a string of amino acids that we identify as an extracellular sushi domain on the GABA BR1 subunit, together in concert with the GABA BR2, it will then cause a reduction in the release of neurotransmitters, which is why you see a decrease in neuronal activity. We get less neurotransmitter release. Now, that's all I really want you to take away from this particular slide. But for those of you who are in research, I also want you to take note of some of the other things that are on this slide, not to memorize, but for you to understand a little bit about this research project. So if you look at this article here, this was actually submitted back in mid-August, and then it was accepted within a few months. If you look at this paper here, the amount of work that they actually had to do to get this paper published in a really high-profile journal like Science was actually considerable compared to the other paper. Even though technically the other paper was much more complex, this paper actually took a long time to be published. So it was actually submitted back in 2017. It was not accepted. It got resubmitted 1 year later, back in September. So that means that they had to do a lot of extra experiments for this paper to eventually get accepted. But once they had done those extra experiments, they were able to publish it fairly quickly. And that's really the nature of how science works. It's not always like you get something, even though it's an interesting story, that it gets published. There were a lot of extra experiments that this group had to publish before they could get it accepted. And you don't need to worry about the GABA C, which is only found in the eye. None of those details, nothing beyond sort of GABA A and GABA B for your upcoming term test. Do you have any questions on this mechanism or anything on this slide before we go on? So this does answer this fundamental question. Why do we have this amyloid precursor protein, and what is it normally doing physiologically when we have this being processed and cleaved? Okay? So one other thing to make note of, just as I mentioned to you before, when you are looking at these individual subunits, so this is the GABA A receptor, similar to the nicotinic acetylcholine receptor, a heteropentamer, so 5 subunits, 1, 2, 3, 4, 5. They're not all the same subunit, so this is where we have the hetero part of it. And they have the same sort of topology, a large extracellular N-terminal region, 4 transmembrane domains. And then if we were to take a look, the M2 region for the GABA A receptor is also the pore lining region. So it's identical to what we saw earlier with the nicotinic receptor. Even though it's a completely different ion, sodium is passing through the nicotinic acetylcholine, chloride is passing through here. It's conserving this mechanism that is commonly found within the nervous system. Any questions on this before we go on, or anything that you're not clear about, or anything that you want to make sure that you need to know, don't need to know now? Yes?
What exactly is a sushi domain?
So we'll talk a little bit more about CUB and sushi domains later, but it's a string of different amino acids that form a very characteristic sort of group of proteins that we can identify in many different types of proteins. So it's a motif if you want to think of it that way. Okay? Any other questions? Okay. So just as a review for you, we have been looking at different types of ion channels, the different properties that are associated with these ion channels over the last sort of week or so. And today we went through in more detail that you need to go back and study and you need to know for the upcoming term tests. So if you know that you're not really great at memorization, now's your opportunity to highlight that, circle that in your notes. Will you be asked to draw this? Potentially, you might be asked to draw the topological structure of the GABA-A receptor subunit or the nicotinic receptor subunit, which are identical to each other, and make sure that you understand why these ligand-gated ion channels are important. But ultimately, we're looking at ion channels. So we took a little bit of a detour to talk about one specific type, the ligand-gated type, but we need a way for charged particles, ions, to cross this lipid bilayer. These channels are always passive. They are always allowing for the movement of ions down their concentration gradient. They don't use energy because if they were requiring energy, it would be horrendous for your brain. You wouldn't be able to process things appropriately. And so that means that we need to have an electrochemical gradient. And as many of you will know, the electrochemical gradient exists because of other proteins that allow for the movement of ions. And I'm going to go through these really briefly with you in the next 2 slides. And what we talked about just before we get to those next 2 slides is that if we were to take a look at these different ion channels, and we did that right now, that we have different regions that regulate the binding of neurotransmitters or binding of different elements, how these will open. And we talked about how M2 domains within the subunits rotate and then open up the channel. And we talked very briefly about gating, and we talked about how we can block a channel if it was open. And we're going to come back and talk about gating of channels a little bit more. So of this last bullet point, we really haven't spent too much time on the gating of these channels or their regulation right now. What we have been spending time with is these first 2 bullet points. And so again, this is mainly for your own information. I'm not going to ask you a question this year on this particular slide. How do we set up this electrochemical gradient? How do we have a difference across this lipid bilayer for these different ions? One of the reasons why we have this difference in ions is through the activity of this sodium-potassium ATP exchanger. So this allows for much higher levels of sodium outside the cell, and it also allows for more potassium inside the cell. So that's setting up that gradient because channels only use gradients, and this is one mechanism by how this all works. And again, an ATP-dependent process, so this is ATP-dependent. And another one of these also involves co-transport of different ions as well. So once we have things like potassium, and we have a lot of potassium inside the cell, moving it against its concentration gradient would actually favor, for example, the co-transport of chloride out. So we really are interested in setting up chloride outside of the cell, and this co-transport of potassium and chloride allows for the chloride gradient to be established as well. And when we get into stroke a little bit later on, we'll talk about how some of these different co-transporters, like the potassium chloride co-transporter, actually doesn't work as effectively when we get into disease processes and neurons a little bit later. And again, these 2 slides here, the sodium-potassium ATP exchanger and this potassium chloride co-transporter, these 2 slides you don't need to concentrate on in terms of the details. I'm not going to be asking you a question this year on these slides. Just to make sure that you understand, we do have an electrochemical gradient that is set up by these other types of proteins that we're seeing here. Any questions before we go on? So what we've gotten so far is that we have a lipid bilayer. So we have different regions in this lipid bilayer that are known as lipid rafts. They're specialized. and we took a little bit of a tangent. And then we realized that the only way that ions can cross down their concentration gradient is by having specialized channels. And this is what we've been concentrating on over the last sort of half lecture today and in the previous lecture. And so if we buy into the fact that if we have an electrochemical gradient, there's a difference in concentration across this lipid bilayer, and the only way that these ions can cross is through these open channels that can be opened and closed in different ways that we haven't talked about yet, then we should actually be able to measure these changes. And people had thought about this for a very long period of time. And this is slightly out of order because their Nobel Prizes were awarded back in 1981. But Erwin Neher and Bert Sackman actually thought a little bit about this. And they thought, how can we actually measure the movement of ions? We should be able to, because each individual cell, we know that there is a concentration gradient that exists. We know they have these lipids. And they won the Nobel Prize in 1981 for their work on developing what is known as patch clamp electrophysiology. And so for the next little while, we're going to look at patch clamp electrophysiology and how this technique works. So again, this goes back to beyond what Neher and Sackman had done. Earlier results and earlier experiments, much before they started to think about this, had suggested that you could actually measure a difference across this plasma membrane. This is a different technique where we actually take an electrode, we basically spear a cell. The electrode actually physically punctures the cell and sits inside the cytoplasm, and then we have a reference electrode that sits outside of the cell completely. And when they did this, and when scientists had done this, and they had done this for a very long period of time prior to Sackman and Neher, they had observed that when they puncture a cell and they compare it to the normal electrochemical solution that exists outside of it, they always measured a negative value. So almost every cell that you look at, with very, very few exceptions, if you were to do this experiment, you would always see that it's always more negative inside the cell than it is when it's compared to the outside solution. So people had known this. Erwin Neher and Bert Sackman had known this, and they wanted to know what accounts for this. Can we actually measure some of these different movements of these ions that might contribute to these differences? So this gets into electrophysiology, and one of the most important things that exists within electrophysiology is we have a different type of electrode. And again, apologies on this, but I'm going to go back. This would be an electrode, and we can also have the ability to either push ions in to this cell through this electrode, or we can monitor things that are going on directly through this electrode. So we can actually manipulate what's going on here, and we can also measure things that are going on here. And this means that we can actually hold this membrane potential difference, the difference in ionic charges across this membrane. We can actually control it and hold it or clamp it at a certain level that we are measuring. And this gets into a different type of electrophysiology. So we can clamp the membrane, the difference across the membrane due to the differences in the number of channels or the differences in the concentration of different ions at any point in time because of the electrode that we are using. And because of the way that we have it hooked up, we can also measure the movement of ions, which we're going to call an ionic current. Any movement of ions is an ionic current. Okay, and you can actually see here, this would actually represent an electrode. It's slightly out of focus because the microscope is actually focusing on the cell bodies, and the individual here is trying to get the electrode down to a single neuron and to attach this electrode to this neuron. So I want you to think a little bit about this. What do you think is bigger, the electrode or the neuron? So the electrode is much, much bigger than the neuron is. The neuron, in comparison, is very, very tiny. And I need you to keep that in mind when we go through this in the next few slides. So do you have any questions about any of this before we get on? No? Okay. So again, this is a really busy slide. I don't need you to take away anything from this slide. It's just to give you an overview. You need to know nothing from this slide. I'm going to use the next 2 slides to go through this in a little bit more detail, but you do need to understand how these are all sort of occurring. So on that previous slide that I just showed you, that discussion that we had about the size of the electrode versus a cell, what you're seeing here is how an electrode can come down onto a neuron using a microscope and how it can directly attach to the cell membrane of this particular neuron. We are not trying to pierce the neuron. That was done a long time ago. Those are known as sharp electrodes. Those were done in the 1950s and 1960s. Even though some researchers still use the impaling technique where you physically stick an electrode inside a cell, most neuroscientists and most electrophysiologists do not use that technique anymore because it's very damaging to the cell. So they will use these different electrophysiology techniques that you are seeing here. And we'll go through each of these in turn. And what you're seeing here is in slightly more detail how the electrode is getting closer to a cell, how it will physically touch the cell, and then what happens next determines like what type of recording that we get. Once you've touched the cell, you can decide to make a really tight seal by using suction, or you can decide to actually rip off a piece of the membrane. But these steps here are the ones that actually lead to everything else that follows. What's really exciting now, and again, nothing on this slide is anything that you need to actually worry about. What's really exciting now is that new techniques are being developed for automated, like you don't even have to do this manually anymore, for automated patch clamp. Just using a microscope and using cells that are grown in suspension, you can actually do this very, very quickly with very little sort of technical expertise. The only problem is doing this type of patch clamp, which is identical, so the electrodes are now sitting underneath, and the microscope is up here. It's actually much more expensive, and there are very few labs that actually have the ability to use this automated patch clamp technology. So of the 60 labs here at U of T that use electrophysiology, how many do you think actually have automated patch clamp? Take a guess. 2. Good guess. Wrong guess. Any other guesses? 105. It's one of those answers. What do you guys think? 1. So it's actually 0. No labs here at U of T actually have this technology yet because it's really expensive. And it was actually pioneered down in the United States because they're looking at very high throughput. Like they want to look at thousands of different neurons in a single day. How many do you think you can measure in one day if you're like a really good electrophysiologist? So I was never a really good electrophysiologist. I was just okay. How many do you think a really good electrophysiologist could do in one single day? Assuming that they're not working 24 hours a day. So just a normal regular work day, you show up at 9, you leave at 5. How many do you think you can do? If you're really good. Yes? 60. Wow. Okay, that would be amazing. You'd be like the most sought after electrophysiologist of all time. If you're really lucky and your cells are in really good shape, you might get through about 10 to 12 cells, and that would be like considered a really great day. Some days, even though you've done things identically, you might get 0. None of those experiments will work, and none of the electrophysiology would work, or the cell membranes just don't allow for you to touch or to seal onto the neuron, or when you do touch it, for whatever reasons, the neuron bursts open on the spot. So it gets to be really frustrating, which is why the United States and Europe have developed these automated systems to allow for a much higher throughput. So I'm going to take you through these different electrophysiology recording configurations. And again, it's up to you. It's highlighted in a little bit more detail in this free platform. And some of the students from last year's class actually helped to write that particular chapter. But what we're looking at here is taking an electrode, using a microscope to visualize that we are physically touching a cell, and we are coming right up against it. And what we're seeing here is that when you touch the membrane, and the membrane is denoted for you in red, that we will have a patch of membrane that might have these different types of channels that are associated with it. Now, depending on what we do next, we physically touched a neuron. if we decide that we are going to keep it like this, we have one other option that isn't shown for you here, which you do need to know. If we have physically just touched the cell and we have created a seal, but it's not a very tight seal, we haven't ruptured the membrane, but we want to actually record from the entire cell, what we'll create is something called a perforated patch. And this perforated patch is unique in that we can control whatever fluid is in this electrode. We can control the different ions that are in solution in this electrode, or we can include different compounds that are found within the fluid in this electrode as well. And if we decide that we are going to include an antibiotic, and the most common type of antibiotic is gramcidin, but we could also include things like nystatin, which is another antibiotic, or amphotericin B, which is a different antibiotic, or gramcidin, then if we have that antibiotic within the fluid of our electrode, it will actually create antibiotic pores. It will physically punch holes into this membrane. Okay, so this is a way for us to artificially connect the contents of the fluid within this pipette, this electrode, with the intracellular contents of this cell. So again, I just mentioned a whole bunch of things. Do you have any questions on that before we go on? So this is known as a perforated patch. It's also known as a gramcidin patch. It is a form of a whole cell recording. We have the whole cell attached to this electrode, and we can measure everything, all the channel activity on that particular cell. Any questions before we go on? No? And again, this is with like mild suction. If we decide that we want to take things a little bit further, then we can actually have this situation here, but instead of just stopping here, we can actually increase the amount of suction that we have, and it will physically break the membrane. And the membrane will actually make a really tight seal because it's lipid. It will make a really tight seal with this glass electrode, meaning that here now we have what is known as a whole cell recording. We are also, again, in contact with all of the different ion channels that are found within this particular cell. So you have to think a little bit about this, and you have to think very carefully about this. Is this whole cell recording? It is. This is a type of whole cell recording. Why might we want to have this type of recording versus this type of recording? Because aren't they really the same? Aren't we really just recording all of the channels from this cell, and aren't we doing exactly the same here? We're just punching holes within that. So the antibiotics will actually dimerize, form a physical pore within the lipid membrane, but it's basically like having access to the intracellular compartment. Any ideas? So here's the major reason why we want to sometimes do this perforated patch. If we're looking at the movement of small ions, and we only want to look at small ion movement, these perforated patches are much better at maintaining some of the different electrochemical gradients if we want to look at it under native conditions. Here, because you told me, and you were 100% correct, the actual pipette or the actual recording recording pipette is much, much bigger than the actual cell itself. It actually will completely swamp out or replace the intracellular contents of what you see here. These become incredibly quickly equilibrated, and so it becomes harder to look at what is the normal sort of concentration of different intracellular molecules, etc. But having these different gramcidin patches allows us to keep a little bit more of the integrity of the difference across that plasma membrane. Okay, and I think, let me just take a look at the time. I'm just going to go through two other things really quickly here because they'll be important, and then we'll take a break. So under these different conditions, if we have made contact and we are physically now connected with the plasma membrane directly, if we were to pull away from the membrane, so either with mild suction or after we've made this really tight seal that you see here, we can actually rip away a little bit of the membrane, and some of those different membranes will actually have channels that are associated with them as well. And depending on how we do it, and there's some more details on the next slide, we can have different configurations of the channels that are within the membrane. So we can have what is known as an inside-out recording, and here the overall structure and topology and the way that the different channel is organized is kept intact. It's still in exactly the same configuration that we would normally see. And here, if we were to pull away, after strong suction has occurred, the membrane will actually seal itself up again, and we will have what is known as an outside-out recording, where we have the outside region of the channel or the receptor being exposed to the outer parts of the extracellular solution. We're not going to talk about these two conditions today or immediately, and we're just going to be talking about patches in general, but I wanted you to be aware of these two different configurations. It's probably a good idea to know like what configurations these are. And again, these allow for the analysis of single ion channels, which is what these represent. We can see one channel at a time, which is what you are seeing here. So both inside out and outside out patches allow for you to record one channel at a time. And again, you don't need to worry about the details of do you need high calcium, low calcium. I just want you to be familiar with how the receptor looks in an inside out versus an outside out patch on the term test, because you'll need to know that a little bit later in the course, and I want us to have that same common language. Do you have any questions on anything before we go on, or any questions on any of the previous slides before we go on? Okay, so if you're okay with that, maybe we'll take a 10-minute break now. We'll come back in 10 minutes. I know that there was a lot of material, so think about it, ask questions, and then we'll come back in 10 minutes. If you did not pick up a sort of orange sheet, please do so. There are some extra copies up here at the front. We're not pumping in ions. We're using an antibiotic that we're keeping in the recording pipette to be able to make these different.
Sorry, we're using an antibiotic
To create these pores. And that allows for ion circulation. So antibiotics like gramcidin or amphotericin will actually dimerize. They'll form the inner part of a pore, and then they'll form the outer part. And as they dimerize, they actually create a channel that will go through the entire membrane. So that gives us access to the entire cell and all of the contents of the cell, and it allows us to record from all of the different channels that are found on that cell that we're recording from.
Oh, sorry.
So, Professor,
Thank you.
you said that the perforated patch is a form of wholesome recording
It is.
that can measure all ion channel activity, and it also is used to maintain the electrophysiological.
The girl next to me. Thank you.
Sorry?
Is it also used to maintain the electrophysiological measurements?
Nope. It's there to keep it more physiological than anything else, because we're not dialyzing or diluting out the normal intracellular contents.
Okay.
Thank you.
Okay, sounds good.
Thank you.
I'm just kind of confused about this. It's kind of hard for me to visualize how this forms this. So I was thinking maybe M2 and M4, they kind of form a circle, which forms the inner wall of the channel. I'm not quite sure if that is correct.
Well, you can look at it. Assuming you need more than one. Yep, think of it that way.
So does that mean M4 is also part of the inner wall?
Not when the channel itself is open.
And also for the last
Not too bad, not too bad. This class is so good.
for the inside out
Yeah,
recording,
it kinda
that's
stresses
only,
me out.
that only happens for the miles.
I know, I can't sit in the same spot every time.
So if I'm physically
Kind of mad. I
disrupting
kind
the
of like having my own
Yeah.
self-assigned
cell
seat.
membrane,
And I get displaced and I'm like stressed. No, but I really don't have time to like come through. No.
physically adhere to
Who has time for
my
that?
recording pipette, then that's like where I form what's called a giga-insular, which are the two.
I can also get to. Thank you.
Do you need any?
Yes, please. Yes, sorry. Thank you.
I just had a question about the city, guys. Is this communication going to be through group chats or is this?
So this week, Delphine, who was up here, and Marlene will be doing this template. If you want to organize a group too, she communicate with them. They have a template. They know sort of what they need to do. I'll just check with you. That's easy.
Okay, all right. So we have to just contact our group members.
So are you on the Facebook group? I think you're
Yes.
on the Facebook. So I would just maybe connect with 4 or 5 minutes. I would maybe
Like
also
Amelia.
whoever you know.
Right.
If you know them, great. We just introduce them.
Can I? Thank you.
For the GABA A, and down here underneath the GABA B, there's a for the GABA.
Okay, but are these representing like one of these cylinders of the different subunits, or okay, this is a full GPCR. So okay, but these ones, the alpha, it's like the acetylcholine. So these represent like the different subunits. Oh,
So, so this would be like an actual GPCR, which is 7 transmembrane domains. So if you look, there's 7 transmembrane domains versus like the GABA A receptor, which has it's a heteropentamer, so you should only see 5. And each of those consists of an alpha or a beta.
whereas you have an R1A GPCR and R1B. Are you still looking for people? Just in case someone needs a
I think if you want to go talk to Marlene and Delphine, do you know them?
I do.
So just ask them to add you to the Facebook group or talk to Kate and one-on-one.
Just had a question about the configuration. So we saw that this was the side of plasmic, that's why it's like inside out recording. But then for the outside out, we don't know how this it touches the extracellular.
So it's going to be really complicated, but if you find a tight seal first, and then you retract that thing and then things that happens is you drag along the long needle. So you actually want to know what it's like. So this is what you're seeing here is the real process, and in that case, the inside part becomes the outside part. So I have to try to figure out what sort of visual analogy for Wednesday. I'll work on that a little bit. But if you get a long enough piece, and whatever was originally on the inside and this is outside, and now when it kind of moves around, the outside part will sort of be a different configuration. It'll be different. So you'll have access to different parts of it.
So are they measuring the part that's touching the inside of the alpha?
No, you'll still measure across this channel. That's the only way ions can flow is through channel. That's still like the basic principle.
But it will be called the extracellular matrix instead of just because of the flare.
I think that
And also for the perforated mesh, are you also measuring like the activity of a single ion channel as well?
You're not looking at a single ion channel. You're looking at you're basically recording from every single channel that's on that cell.
Oh, so from in that entire zone, every single.
Nope, not that entire zone, that whole cell.
Okay.
It's like literally taking a snapshot of like everyone in this room. You're not concentrating on one student, like looking at the whole room at the same time. That's the whole cell. Any of the whole cells look at all of the activity of all the channels that it's physically coupled with on that particular. Talk to like Kate or Marwan and ask them to add you to the
Okay,
Facebook.
thank you.
Okay, thank you for the questions. They were really good questions, and I know that some of you are having a hard time trying to visualize like how these different things are moving to allow for channels to open, etc., or what these different inside out, outside out patches are. So I'm going to try to find some resources to help you visualize that a little bit differently than how I've explained it to you in lecture. Or if you have any ideas, or if you have your own analogies that you are able to develop, let me know. I'll try to get those for you for Wednesday's class. So I want us to kind of move beyond that to actually starting to think about why we are making these different types of recordings. So here, and again, you don't need to worry about, is this an inside out, outside out patch? That's not really relevant for these discussions. I just want us to think a little bit about this for the time being, because this gets into some very good conceptual ideas and how we can actually biophysically predict as well as measure things in neurons or in any cell that has channels. So the first thing is, going back to what we've been talking about, I can pull a patch of membrane, and that membrane patch will have channels. It still consists of lipids, and it still consists of proteins, and the only way the charged particles can move across this membrane is through any of those proteins that happen to be open and that it can actually flow through. That's still a basic fundamental property of electrophysiology. So I will have, for example, a patch of membrane that is shown to you here, and that little patch is over here in the circle that you have highlighted. This patch of membrane contains one channel that's spontaneously opening and closing. So this will mean that only potassium ions will flow through this particular channel. If you understood what I was telling you previously, one of the things that makes electrophysiology incredibly useful is that as I connect with a cell, or as I start having these different electrodes, one of the things that I can do is to control what is found in the electrode that I am using. In this particular case, I can control what is found inside this electrode, and I can control the amount of potassium that is found within this electrode. So I will have, for example, 150 millimolar concentration of potassium inside this electrode. The other thing that makes this technique incredibly useful is because I have it connected to a number of different apparati, I can control how much membrane potential differences can exist. I can control, for example, the amount of voltage that I can push through this electrode across this membrane. So again, just to orient you a little bit, I can measure, intracellularly, I have 150 millimolar, and I have outside in the bath that it's sitting in, this patch of membrane is sitting in a different bath, the concentration of potassium outside is 150 millimolar. So if I told you that the concentration of sodium was 150 millimolar, do you care if we're looking at the potassium channel? Thank you. Awesome. So potassium is only going to flow through a potassium channel. Potassium channels are the only thing that I'm recording from, and that's, again, another important thing to keep in mind. So if I were to take a look, and I can measure these different changes that occur. Right now, at rest, when it's not doing anything, and I have this one potassium channel at rest where I have pulled off a patch, I have controlled the intracellular concentration of potassium, and in the bath around it, it's 150. There is no concentration gradient. One of those fundamental properties that we talked about earlier, neurons and channels do not require energy. They require an electrochemical gradient. One gradient does not exist right now. The other thing that I told you is if we get the movement of ions, then we are measuring ionic current. So right now, if we were to also measure from this particular electrode, we don't see the movement or net movement of ions. There is no current, which is on this y-axis. So on this y-axis, there is no net current, which is why it's sitting at 0. So it doesn't mean that ions aren't moving, but there's no net movement. There's no concentration gradient for it to pile up on one side versus the other. They might be moving back and forth, but it's an equal exchange because there is no driving force. There is no electrochemical gradient for that to occur, which is why we have this 0 current. So 0 current means no net movement of ions. Is that all clear for everyone before we move on? Okay. Now, what would happen, or what might happen, if suddenly, across this membrane, I now introduce a voltage change by directly injecting in voltage through this electrode? And again, I won't go into the biophysics of this. You'll have to take it for granted that I can physically change the voltage across this membrane. Up until now, it's been 0. And now, across this membrane, I actually start to increase the voltage so that it is higher outside than it is inside. So I have a voltage change of plus 20 millivolts across this patch of membrane that I'm recording from. If you were to take a look at this now, do you actually see movement of ions based on what you see in C? Yes. So I actually see the movement of ions out of the cell. Okay? So if I apply this voltage difference across the membrane, then I actually see the movement of ions across the cell. And then the same thing, if I were to suddenly make it more negative inside with respect to the outside environment by actually causing a negative voltage, minus 20 across this membrane, I will also see the net movement of ions into the cell. The only ion that we're looking at is the movement of potassium ions. So it's only potassium that's moving in and out through this particular channel. So I can change the voltage. I am experimentally manipulating this voltage here at 0, at plus 20, or at minus 20. And I am measuring the response of what happens in terms of the net movement of ions as a result of the change in the voltage that I am changing using my electrode. Is that okay for everyone before we go on? Okay, so when we do these types of experiments, one of the things that we can generate, and this will be very important because you will be asked to demonstrate that you understand what an IV curve is, and this is a very artificial type of environment. So I want you to be very clear. The slide that you see on slide 20 and 21, although we're talking about potassium, is very different than the potassium that you're going to see on slide 31 a little bit later. Why are they different? The conditions for these experiments are completely different. Right now, there's no concentration gradient. I am physically manipulating the voltage change across the membrane. So it's at 0 millivolts or at plus 20, and I'm physically making this change across the membrane. And as a result, I am going to be able to, by plotting out each of these, so 0 millivolts, I see 0 current. At plus 20 millivolts, I see a net plus 2 millivolts, plus 2 picoamps, I can actually start generating what is known as an IV curve. So I can physically map this out by changing the voltage, measuring the current that results from this particular channel, and I can generate what is known as an IV curve. And this shows me, for a particular channel, what the current voltage relationship is. So based on the shape of this curve, what is the shape of this curve? It is a linear response, right? So it's a straight line, and some channels will be linear like this, and some channels will not be linear. Okay, so just some basic concepts and basic things for us to be aware of. Any questions on this before we go on? Okay, so again, these different arrows just show you how we mapped all of these different things out. So for this particular IV curve, for this particular ion channel, and each ion channel has its own IV curve, if we see this linear response tells us that this channel, and only if it's linear, or only in the parts of the curve where it's linear, a straight relationship, or a relationship that is a direct relationship, as voltage increases, current increases, or as current increases, voltage increases, it will follow what is known as Ohm's law. This is very important for you to keep in mind. So any linear channel, which is what we're looking at here, will always follow Ohm's law, which is mapped out for you here. Any questions on this before we go on? So this is a linear curve. This is therefore following Ohm's law. That means that this channel, we can apply Ohm's law to this particular channel. If it was not linear, we would not be able to do this. Okay, any questions before we go on? And you've all heard of Ohm's law. It might have been a while since you've done first-year physics or anything related to voltage-current relationships. But this is sort of a linear response. It follows Ohm's law. The other thing to keep in mind, we can rewrite Ohm's law. And because we're looking at a membrane, and because the only way that ions can pass through this membrane is through open channels, we can look at rearranging Ohm's law, and we can isolate for current. And by dividing voltage by resistance, one factor that comes up, the inverse of resistance, is known as conductance. So that means that the number of channels that you have will directly affect the current that develops, which makes sense. So the more channels you have, the more ions will flow through. If ion movement equals current, it makes sense that it will be directly related to this. Okay, so a rewriting of Ohm's law is where we have the current equal to the conductance times the voltage. And this channel conductance is, are the channels open? How many channels do we have? Etc. So this gets into this idea that conductance, this value G on that previous slide, is kind of related to what you typically call permeability. So the conductance of an ion channel in a patch of membrane that we're looking at is really dependent, again, sort of in the same sense, on the permeability. If it's open, will ions actually pass through, which is what permeability means. So it depends also on if there's a concentration gradient for these different ions, even if a channel is open. And again, I'll get to that in a moment. So an open channel increases the permeability of the ions. And again, it's a technical term, but if the channel is open, and therefore if the channel is permeable, technically, if we have an open channel and it's permeable, and ions are actually moving through that channel, then we have what is known as conductance. So that's why when you learn about it in physiology, we use those terms interchangeably, because most of the time, under normal physiological conditions, if a channel is open and it allows ions to pass through, they will. And so therefore, an ion channel that is permeable is, under physiological conditions, also conducting ions. Okay, and it's just a technical term. I'll use those two interchangeably all the time, but I wanted you to be aware of it. You are not going to be responsible for being able to distinguish that like on a multiple choice or anything else. So I wanted you to be aware that what we looked at earlier is actually not physiological. That is not something that normally occurs. We normally never have equal concentrations across a plasma membrane. So because of the sodium-potassium ATPase, because of the co-transport systems, usually for different ions, particularly the ones that we're interested in, we have a difference across the membrane of these different ions. So the ones that I told you earlier that we're interested in are things like potassium, sodium, chloride, as well as calcium. And the ones that we're primarily interested in over the next week are potassium, sodium, and chloride. If we look at the intracellular concentrations, things that are found inside the axoplasm, and that's just the axon, so the intracellular contents of the axon, as you likely will recall, it's much higher inside than it is outside. So again, whether it's seawater or blood, this would represent the extracellular compartment. And sodium is much lower inside the cell, or in this case, the axoplasm, than it is outside. This examination of the axoplasm is going to be important next week, which is why I'm bringing this up here. You do not have to memorize these values. It's just to illustrate what you've learned previously is still applicable and why you've learned it the way that you have. So high potassium inside, low potassium inside, and it's really high outside the cell. And for chloride, it's the same, high outside the cell, low inside. And for calcium, it's exactly the same, very high outside the cell and very low inside. Again, this is several orders of magnitude. These are millimolar. This is in micromolar. So it is a very high concentration gradient for all of these different ions. So this means that if we were to look and do a recording across a normal membrane, not that artificial thing that we looked at in slide 20 and 21. So we pulled off a patch of membrane. I kept the potassium concentrations the same. But now if we look at a normal membrane, what we normally see physiologically, we won't actually see what we saw on slide 21. We'll see something very different. So the resting membrane potential that we are aware of is where we have this steady baseline value across the cell membrane, especially of neurons at rest. Doesn't have to be a neuron, but that's what we're looking at in this course. And then we can change this resting membrane potential by looking at the different parts of the ionics, the different ions that are contributing to this resting membrane potential. Okay, so again, keeping in mind on that previous slide, much as you've learned before, potassium is high inside the cell, sodium and chloride are high outside the cell. This is help us to understand this resting membrane potential and how they contribute to this resting membrane potential. So let's take a look at each of these in turn. So again, we're looking at this ionic equilibrium. We're looking at normal real-world electrophysiology or real-world conditions and how we understand this from an electrophysiological standpoint. So if we look at the potassium ion, and we know that this gradient where we have higher amounts of potassium inside the cell, I showed you that as an axoplasm previously, and outside the cell, either seawater or blood, we see that there is already a gradient. So will this gradient stay the way that it is on this particular diagram? Will I see a current on this diagram? Again, current meaning the movement of ions across a membrane. Will I see that on this diagram? Yes? How many of you are nodding your heads yes? If you are, please stop. Okay. What do I need for ions to move across this membrane? I need a channel. Positive charges cannot cross a membrane. Never. Like, they cannot cross. Even though a gradient exists, that gradient will stay there until some kind of channel across this lipid bilayer comes into existence. It will stay like this forever until a channel exists. So right now there is no channel. If I put a channel in there, now there is a way for a positively charged ion to move across this particular lipid membrane denoted here. There is an electrochemical gradient, as I mentioned to you earlier. These are all passive properties. So this gradient exists. We have the movement or the net movement. So we should be able to measure a current. So if I were in patch clamp mode, I should actually be able to measure a current that actually exists. So the question is, though, as is outlined here, why doesn't this continually move until we have an equal number of potassiums on the left and on the right? So it's not just concentration gradients that we have to worry about. What's the other thing that we have to worry about? So one, do you agree there's a concentration gradient on this slide that we're looking at, slide 27? Agree? I would also agree. We have a concentration gradient. And if we were only looking at a concentration gradient, then they should actually equalize over time. The problem is the molecules that we're looking at are charged, and these have some electrical properties that are associated with them as well. As the potassium starts to flow out of the cell, as it moves along its concentration gradient, some of those positive charges will start to accumulate. And again, it's a little bit disingenuous, and it's not quite accurate to talk about these ions actually don't move very far from where the channels are located, but we kind of use that as an excuse to say that it accumulates very close to where they're actually leaving. So potassium moves out, and it's going to start piling up a little bit around where the channel is open. So this means that we're going to actually have positive charges building up out here, and we're going to leave a net negative charge inside, and that means that we have a potential difference, a difference in charge across this particular patch of membrane. So we'll end up with something that looks a lot like this, which is what the resting membrane potential reflects. And again, like charges repel, it also means that we have to keep in mind that as we get more and more potassium outside, it's actually going to stop it from moving down its concentration gradient as well. So this means at some point, even though they're not an equal number of potassiums on this side and on the right, that because of the differences in charge, that they will eventually enter into an electrical gradient equilibrium. And this difference in chemical composition and concentrations and electrical charges and the gradients that exist there are what we call an electrochemical equilibrium. And this is the equilibrium for each of these ions, like the potassium ion. Okay, I know you've heard this in other lectures, but do you have any questions on this before we go on? No? So if we were to go in and take a look at the equilibrium potential for a particular ion, in this case it would be for the potassium ion, this is the potential across the membrane that would exist. How about we leave that question till after, if that's okay? So if there's an electrical potential that balances the potassium concentration gradient, this would actually represent the equilibrium potential for that particular ion. At this value, there is no net flow of ions at this particular potential. So ions might still be moving, but we don't see a net movement out or a net movement inwards. It would completely balance itself out at this theoretical or equilibrium potential value. And we can measure it for each particular ion that we're interested in. And again, I have to apologize. Earlier I told you this was slide 31, but it's actually slide 32. So if you go back, what's the difference between potassium on a patch of membrane from slide 20 and 21 and compare it to slide 32? Here's where we are actually looking at what occurs in a real neuron. And if we took a patch of neuron and we were to measure the changes in the current, the flow of ions at different voltages across the membrane, this is the type of IV curve that we would actually generate. So the first thing is, and I want us to take this really slowly because I need you to understand this, because you need to demonstrate that you're going to understand this on your upcoming term test. Looking at the IV curve that you see here, is this a linear response? It is not. So I want you to think a little bit about this because I told you this earlier. What is a typical type of channel that creates a linear Ohm's law response or a linear IV curve? Oh, I didn't tell you. Sorry, I didn't tell you actually. So this is sort of what we typically see. A linear response is what we see for a leak channel. It's always open. It doesn't matter like what changes that we see here. Here, it is not a leak channel. It is not always open. Sometimes it's open and sometimes it's closed. And I'll get to that a little bit later. So it's not linear, so it's not a leak channel, but it is still measuring the movement of potassium. So it's a very different looking type of channel that we're looking at here. We're also not looking at equal concentrations of potassium across the membrane. Here, we know that the intracellular concentration of potassium is 90 millimolar, and outside, because we can control this, it is 3 millimolar. So we're matching what we see under normal electrophysiological conditions. So again, let me ask you one other question. So where on this diagram do we have zero current? On this figure over here on the right, or just by kind of cheating a little bit by looking here on the left, at which voltage do we actually have zero current? In other words, if you're looking at the y-axis, which represents current, where is it zero? So at minus 75, we have no current. And that makes sense if we go back to what the equilibrium potential represents. For a particular ion, that's where we have no net movement of different types of ions. Here we're measuring it for potassium. This is actually really useful for us to be able to know. So again, how did we generate this IV curve? We took a patch of membrane, just as I showed you earlier. And by changing the voltage across this membrane, we just measured like what direction is the current moving, what is the size of the current, and by lining up each of the points, we generated this type of nonlinear curve. And this curve is nonlinear, and we look at the shape of this curve, and what this curve tells us is that this channel that is moving ions is rectifying. And again, I'll get into that in some more details later. You are not responsible for identifying whether it's an inwardly or outwardly rectifying channel. I just need you to know the difference or to be able to identify. If I see a nonlinear curve that has a different shape to it, that these are opening and closing more in different voltages than in others. Okay, so this is clearly a rectifying curve that we're looking at. And much later on, next week in Monday's class, you'll be able to tell me like why it is a rectifying channel and why it's not a leak channel and what contributes to it being a rectifying channel versus being a leak channel in Monday's class. So why are we worried about all of these different types of things? So again, let me make sure that we're all on the same page. When I'm looking at this, am I physically measuring this? Or am I calculating this as a theoretical value? So this plot here. So I am taking a patch of membrane. I'm controlling the intracellular, extracellular ion concentrations. I am physically measuring this myself, okay? So that's one thing to keep in mind. I'm measuring this using electrophysiology. Another way that we can do this is by theoretically calculating some of these different types of electrochemical equilibrium potential values. So the Nernst equation, the one that you are the most familiar with, actually allows us to theoretically, because we know the extracellular concentration and the intracellular concentration, and we know how these work for different ions and the charges of these different ions, and by keeping these are always consistent or kept constant, by using this value for RT over F at 25.26, we can actually calculate a theoretical equilibrium potential for each of the ions as long as we know the extracellular and the intracellular concentration. So is that part clear for everyone? Because I'm going to make a big sort of connection in a minute. So we can measure it theoretically, the Nernst equation, or we can actually go in and measure it using electrophysiology, which is what we've been looking at with IV curves. You need to understand those two things first before I start linking them together. Is that okay for everyone? Yes? Does the number of channels matter for the equilibrium potential? It does not. As long as there's a channel, that's what we're calculating. Okay? So I told you previously that when we do these sharp electrodes and we physically impale a cell, as soon as we impale the cell, so up until this point in time, so I'll look at 0. So I've kept this electrode outside, it's at 0, it's exactly the same potential difference. There is no potential difference, it's measuring exactly the same thing when the electrode is outside of the cell. I move it closer and closer, and right here, as soon as I insert the electrode, I immediately, compared to the outside environment, measure a negative resting potential. As soon as I impale this cell, now this electrode is measuring a different ionic environment than the electrode or the bath electrode that is sitting outside of the cell. And that's why we get this negative resting membrane potential. And it's always this negative value. In neurons, even if it's minus 90, minus 65, minus 75, it's always a negative value that we are measuring. And so going back over the Nernst equation, we can start to see how different ions, because we know all of the different ions that are involved. We know that there's sodium, potassium, chloride, and calcium. If we start using the Nernst equation, we might be able to start modeling why we get this negative resting membrane potential. So is it due to any one particular ion? So if you do the Nernst equation for each of these different values, so for potassium, the Nernst equation tells us that the resting membrane potential due only to potassium would be minus 86.6 millivolts. Is it the same or not, etc. And if we measure this voltage relative to the outside, we don't actually get this value of minus 65. And when we are thinking about this, we have to understand why there is this difference. It's actually a really important difference if we start thinking about this. So we can do this in two different ways. We can do this electrophysiologically, or we can start doing this conceptually. Conceptually, if we were to map out the different channels, we would actually not be able to come up with this resting membrane potential. We could try to work this out mathematically, all theoretically using Nernst, by looking and plugging in different numbers, but it actually doesn't work for any one particular ion. So if we then start going in and measuring this using electrophysiology, we can actually get a better appreciation of what is the theoretical value, what is the actual measured value, and how are these two things a little bit different. So I wanted you also to be familiar with this concept as well. We have talked about an equilibrium potential where we get zero movement of ions, and typically we talk about E potassium or E sodium or E whatever ion we're looking at. And then we can also measure this current, which is known as the reversal potential. These are usually the theoretical and the actual measured values are identical. So I'm going to use these two terms interchangeably all the time. And I'm going to give you some examples for you to work on over the weekend so that you're more comfortable with it, so it becomes second nature to you. As long as you're taking the time to do it, I promise that this idea and concept will become really easy for you to understand, that the reversal potential that we measure in a recording or the theoretical Nernst equilibrium potential are usually equal for a particular ion. And this helps us to understand where all this is occurring. All of this will become important when we talk about the resting membrane potential using electrophysiological approaches that Hodgkin and Huxley, as well as John Eccles, helped to define back in the 1960s. And we'll be talking about how these squid giant axons tell us a little bit about what's going on in these different systems. But you need to keep clear reversal potential and equilibrium potential. Nernst equations, equilibrium potential, we hypothesize or we can calculate. Reversal potentials are what we actually measure in electrophysiology. And we're going to start using the same model systems that Hodgkin and Huxley did when they won the Nobel Prize. So why do we talk about these different values? So the first thing is these squid giant axons, and again, these are just normal squids. I don't know if you guys ever have seen a squid. They're fairly small, but their axons are actually really, really large. These are not squid giant axons or giant squid axons, which again, a lot of individuals get confused on. Giant squids are actually very, very rare in nature. Anything over 5 meters, you don't see them very often. It would not be a really good experimental model, but small squids actually have really giant axons that allow them to escape. They're so large, in fact, that we can physically remove the axon that we can see by eye. It's about 1 millimeter in diameter, so it's easily seen. You can actually physically, like toothpaste, squeeze out the contents of the axoplasm. And so these experiments that were done first by Hodgkin and Huxley were literally isolating the squid giant axon, squeezing out the contents of the squid giant axon, figuring out what made up the axoplasm, and then replacing it with an artificial axoplasm that they created themselves. And they wanted to know if this is a normal action potential that they generated, if they were to create their own version of the intracellular solution, would they generate the same action potential? They did. And so they were able to accurately predict, reproduce, and then replace the axoplasm, which is the values that we looked at earlier. So when we use those values, it's because they literally spent a whole year and a half in their lab squeezing out these axons and then figuring out what were the ionic concentrations, replacing those ions until they got the exact same action potentials that they measured after coming up with their own sort of recipe for the axoplasm, getting it right, and then putting them back into the axon and then trying to generate the exact same action potentials. Again, I don't know if you would have the patience to do that. I wouldn't, but they spent like a good year and a half doing these experiments. So if you were to think about this, this is a lot like what you do when you're worried about your GPA. This is the Hodgkin and Huxley GPA problem. So they calculated that by the end of this course, based on term test 1 and term test 2, all my assignments and my quizzes, I should end up with like an A+. Should have at least a 92. And yet, when they got their final marks back, it wasn't quite that 92 or 93 they were expecting. Turns out that they ended up with like an 85 or an 84. Would you be happy with that? Well, this is really good. So like when it comes to adjusting marks at the end of the semester, this is going to be fantastic. You guys are okay with that? No. So they were kind of flabbergasted. They were able to accurately reproduce, replace this axoplasm, and they were able to do so that the action potentials look exactly the same as a normal axon. They calculate what NERSC would be able to calculate for potassium. They're measuring this resting membrane potential, and there's a difference. There's minus 93 versus minus 65 or minus 75. What is the ion that's accounting for this difference in what they expected to see, which is they were hoping it's all due to potassium, versus what they actually saw, where it's like minus 65 or minus 75. And this is what really led to their ability to win the Nobel Prize. This small gap, this difference of about minus 18 millivolts. How were they able to account for this, and why did they think it was that important? And it turns out, again, we'll talk about this more on Wednesday, the membrane at rest is also permeable to sodium. And that's that small change, even though they didn't predict that this was going to be all that important, was really what made up that little difference that they could account for. And this, again, is really important. It's sort of a throwaway line now, but you need to keep this in mind. If you were to look at a patch of membrane at rest, not doing anything in a neuron, it turns out that neurons are 10% less permeable to sodium than they are to potassium. So based on what you now know about permeability, what accounts for permeability, and the fact that these are all due to leach channels, because these are at rest, they're just open channels, what can you tell me about the number of these channels? Which one do we have more of? So permeability just means that these channels are open and that ions are actually flowing through, and the sodium has 10% of what is there for potassium. What can you tell me about these leach channels? So leach channels for sodium versus leach channels for potassium. We have many, many more leak channels at rest for potassium than we do for sodium. And I want you to keep that in mind. So we're going to be talking about these leak channels, which are basically like open holes. We can't really regulate them. And then we'll be talking about how Hodgkin and Huxley were able to determine, based on these values, what the action potential actually involved. So we've already talked about earlier today about how some of these channels open and close in response to neurotransmitter binding. So we started off today looking at acetylcholine receptors, and then we transitioned into GABA A receptors. And by the way, if you're thinking, I don't really need to know a lot about GABA B receptors, I can tell you now before term test 1 that you do need to know that diagram about GABA B receptors and how GABA B receptors might regulate neurotransmitter release, especially on term test 1. And then we moved into the differences between calculating versus actually electrophysiologically measuring the membrane potentials and all of the different ions that move across this membrane. And in the next lectures, we're going to be starting to talk about the driving force. And by the way, in your notes, it's a little bit different. If you're asked to give an equation for the driving force, this is really the equation that you should be writing. It's not sort of I which is current, is equal to Vm minus Ek. It's the driving force is the membrane potential that you are measuring minus the equilibrium potential that you can calculate or that you can actually measure